Okay. So my name's Guy Coleman. Those who don't know me, I'm a PhD student at the University of Sydney, currently based in Texas, though, doing a chapter of my research over here until the end of the year. And that's with Texas A&M University. Daniel is joining us as one of my colleagues. And you can follow along with some of the research that I do over at Twitter, at Guy Coleman, or also at WeedAI News is where we try and update all the, or keep everyone up to date with the latest developments and data sets and things that are happening with AI. But this is an introductory webinar, I guess, and so we'll work through the background to green-on-green weed detection and then how WeedAI is really addressing that. And as I mentioned, this is part of the second stream, so you are at part A. And I've left a gap between the first and second stream, so you have the opportunity, if you feel like it, to record or collect some images yourself, annotate them, and then also upload them in the second part B. And that way you'll have the opportunity, I guess, to contribute to weed detection around the world. And if you don't have the time, that's perfectly fine. I'll still go through that process with the data set that I've collected. It doesn't have to be anything big, but I'll provide more details about that once we get to the data collection section. And as I mentioned, these will also be recorded. So today we'll go through site-specific weed control, so what that means for green-on-green. Then I'll run you through a live WeedAI demonstration, so take you through the website. And I encourage you during this whole webinar to keep WeedAI open in the background or have it on the second screen. And then you can refer to it as I mention it and follow along on your own computer and play around with it. Also go through image collection. So we'll spend a bit of time looking at the right ways to do that and what to record. Then also image annotation. So once you've collected those images, how do you actually annotate them effectively? And I'll give you a few tips and links on how to do that yourself. And finally, if there's time or if there are questions, we can have a bit of a discussion as well with any Q&A or at the end there. But as I mentioned, this is meant to be fairly informal, so feel free to jump in at any point with questions, either via your audio or also via the chat. I'll keep an eye on both. So to kick things off, the first thing to recognize is why we need site-specific weed control, I guess. For some of you who are agronomists, extension agents, researchers, this will be fairly obvious, but just set the scene. This is up in Narrabri, University of Sydney's farm in northern New South Wales, Australia. This is about 7 hours north of Sydney. And while this is obviously in a fallow situation, you can see there was a wheat crop here. It represents the range of different weed densities that might be present in a crop. So you could have individual weeds like you have here. You might have large clumps where there are dense clusters of weeds. Or over on the right here, there might be sparser clusters or even no weeds at all, as you have on the left. And what that means is when a farmer might spray the entire paddock, well, for a certain weed or to control the weeds there, they actually are only really needing to control a very small percentage and apply herbicide to a small percentage of that field. And that's illustrates, I guess, the wastage or the potential for savings and more targeted options that may be relevant when used on a site-specific basis. As a site-specific weed control, because of this, it is all about treating the weeds and not the paddock. And it's an individual, for those not familiar, paddock is just a field, but basically it's treating plants on an individual basis as opposed to a whole field unit area type treatment. And the benefits are quite profound. We've seen these with weedits and weed seekers, all those reflectance and fluorescence-based sensors since the 1980s when they were first developed. And some studies have shown the reduced inputs by between 40 and 90%. But of course, that's just based on a weed density equation. So the fewer weeds you have, then the less herbicide you'll use. And this is in fallow situations from what I've read. And because you're reducing those inputs, that is herbicide in the spray or spray application scenario, then you can reduce that input and use either new herbicides, potentially more expensive, or also alternative weed control methods that might use quite a large amount of energy, things like lasers, if they're applied on a whole field basis. But if you applied them on an individual plant basis, they might become more relevant. But to do all this, we need weed recognition. So we need to be able to identify where weeds are in a crop to do this effectively. And that's the fundamental component of all this, is that you actually need to be able to detect weeds in all different scenarios to do this effectively. So as I mentioned, we've had green on brown weed detections. That's for green weeds in brown backgrounds or fallow scenarios. And these sensors were first developed in the 80s and 90s, and they just look for difference. They're quite really fascinating, interesting, simple sensors. They just look for differences in fluorescence if they have an active light, or reflectance if not, in that chlorophyll region. And because of that greenness, and the plants typically reflect near infrared strongly and absorb red light quite actively. So if you look at that difference, then you can detect a weed, the presence or absence of a weed. And these work well in low weed densities. There's been fairly widespread adoption in Australia and elsewhere in the world, in South America too, I think, so down in Argentina, and probably Chile, Lorenzo, I'm guessing. Feel free to mention in Chile if there's any use of these. And these examples, I guess, are just two ones that exist, but I'm sure there are others out there. So for in-crop weed detection, in-crop weed detection, we can have what's called green on green sections. So you're detecting a green weed in a green crop. And what we can use for those, and what we need to have that more complex detection, is a more sophisticated approach. So a more sophistication in your data. So not necessarily just your reflectance or fluorescence data in those two spectral bands, but you need images this time, and also more sophisticated algorithms that can handle that data too. So for the sensor-based approach with green on brown weeds, we could get away with the sensors. But as we move to image or camera-based detection, then we can start using the more advanced machine learning algorithms. But the drawback of these approaches is that they require substantial amounts of training data. And I'll get to that. And obviously that's the reason behind weed AI, and I'll get to that shortly. But there are a number of different players in this space. So these are just a few of them that come to mind with an Australian context. So for the sensors, obviously we've got the weed seeker weeders of the world. AutoWeed is a group up in James Cook University in Queensland in Northern Australia. Bilberry is a French company working out of Western Australia, developing green on green, but also, of course, because you can have a camera detect green, you can also use them for green on brown, but there's a different approach there. And then because the camera-based, image-based approaches can then deliver that information, that data stream to more advanced algorithms, then you can start to get those in-crop green on green detections too. And that's where these players are also operating, and I guess that's the differentiating factor. And that's for those broadleaf weeds and cereals, or grass weeds in broadleaf crops. But grass weeds in cereal crops are much more challenging because of those visual similarities. And haven't seen anyone come out and say they're doing this commercially just yet. So there are a range of different weed detection methods we can use. So for those green on green scenarios, we don't necessarily have to use, or they aren't all the same, I guess. There's a different range of different methods we can use to find out where those weeds are. So at the broader scale, we have classification, then we have more fine, precise object detection, then we use semantic segmentation and instant segmentation too. And so I'll just go through each of these in a little bit more detail, because I guess they're fairly important with the range of opportunities they give us on the weed control side of things. So image classification actually tells us if a weed is present or absent in the scenario, in the image. So in this scenario, we have turnip weed. So you can see that the turnip weed's in the top left, but the algorithm just tells us that in this image, there is turnip weed present. We don't get really much information on its location. And I guess this would be useful for spot spraying, where you can have coarse treatments and you're not too concerned about the precision application. And of course, if you're still doing spot spraying, you're saving a substantial number of inputs. Then we have the object detection. So this is an algorithm that provides more information on the location of that weed or that plant. So now we know there are 4 different turnip weeds within this image, and we have the bounding box that goes around each of those weeds. So now we have that more precise information on where that weed's located. So we still could do spraying with this, but we could also do more precise applications of potentially electrical weeding or other alternatives that require much more information on the location of that weed. At the pixel level, so every image is made up of a series of pixels. So if you have an image that's, say, 32 or 3,208 pixels by 2,200, that just happens to be the resolution of one of our cameras we use, then you have millions and millions of pixels within that image. And what semantic segmentation does is classify each of those individual pixels, the millions of pixels, into a class. So either a weed class or a background class or a range of different classes that you give it. And so this gives you then that really precise information on location. But one drawback of semantic segmentation is you don't get those individual weeds, the instances of weed, each of those weeds. So we know that all these red pixels are turnip weed, and we can do a bit more other computing to work out that, all right, this group up here is one group of turnip weed, this is another group, but we don't get those individual instances that you might get with the next method. And this is quite targeted, so you can potentially scan a laser over that, or you can use more targeted options. Finally, we have instance segmentation, and this is potentially the most detail you can get out of a weed detection algorithm. So you have information on the location of the weed, and also the number of weeds in the image, and also the number of pixels belonging to each of those weeds. So highly targeted methods like lasers would potentially work well with algorithms like this. And the main drawback of moving up this scale of precision, as you'd probably expect, is it's more intense computing power, so each of these tends to run slower. From what I've seen, I think these are operating about 8 frames per second, the instance segmentation methods, or up to probably 20, I think, depending on the computer and the size of the image and the algorithm you're using, but they are much slower than, say, the bounding or object detection algorithms that can run in the tens, I think even hundreds of frames per second at the moment, depending on which one you choose. But that's just an overview of the weed detection methods. This is the output we get from the algorithms. I thought I'd also provide a bit of background on where these algorithms actually fit. So why are they better than what's happened previously? And to get a feel for that, it's good to place them within the different terms or I guess names you might have heard. So the first being artificial intelligence. That's often thrown around quite widely. But what it is a very broad group of things that mean systems that acts like humans, I guess, in inverted commas. Basically, they have some form of intelligence, again, in inverted commas, that they just do things that are not hard-coded or necessarily one input equals one outcome. It has a little bit more smarts about it than that. Within artificial intelligence, you have what's called machine learning. And as the name suggests, it means that a program can learn parameters or learn trends or information and patterns in data without you explicitly programming it in. So for example, an AI might be like a color-based weed detector. So you're looking for green, where you say, all right, look within these thresholds for green. But then a more advanced approach might be to give it all the data you need. Say you identify all the weeds in 100 different images, and you say, you find the patterns within those images, and then you set your own parameters, as opposed to me or another researcher or programmer saying, okay, look within these bounds. And so that's how machine learning differs, I guess, as a subfield of artificial intelligence. It doesn't have that explicit programming input. It learns itself from the data. And then within deep machine learning, we have the subfield of deep learning. And that's just a series of different types of algorithms and algorithm structures and architectures. And I guess the technical term is that they tend to be multi-layered artificial neural networks. So an artificial neural network is something that was very loosely based on a neuron in a brain. It was thought of way back in the 1940s by a couple of researchers. And they thought it was going to revolutionize the world back then. And they even tried to build hardware, like physical neural networks, which is quite fascinating, actually, if you're ever interested in reading up about it. But basically, these can represent much more complex patterns within data. So again, still machine learning, you give it the answers and it finds the solution, I guess. But it's just a different way of representing that data. It's just a different algorithm. Just like you might have learned at school that y equals mx plus c, these algorithms combine lots and lots of those simple algorithms into one more complex network. But I won't butcher the explanation. There are many better people that can explain it with diagrams and videos than I can, and I would recommend checking out a site like Machine Learning Mastery or the many different YouTube videos on the topic if you want to find out more. But what's important about deep learning, I guess, is that, and I'll get to that next, is that it has benefits in terms of data usage. Now, before I move on to that, I'll just say that computer vision is all about just trying to derive meaning from images. So it can use artificial intelligence or machine learning or deep learning. It doesn't necessarily have to use anyone specifically, but it does just fit across all those different areas. It's all about trying to understand what is in an image, because humans and animals have a really fantastic ability to see something and then immediately understand what's there because we've had a built experience. So trying to teach computers to do that is a lot more difficult. And that's why deep learning has shown such promise, because it's, I guess, made the most advancement and improvements in interpreting images. And that's why, I guess, most computer vision these days tends to have some form or some part of it fitting within the deep learning realm. So as I was mentioning, why are we so excited and why is there a hype around deep learning? And this slide comes from a guy called Andrew Ng, who's a professor at Stanford, and he's quite a prominent figure in deep learning currently. And if you've ever come across some of his courses on a website he started called Coursera, there are some fantastic ways of learning about the maths and how deep learning actually functions at the very basic level. And he's got some fantastic YouTube videos too. So this is a still from that one of his videos. And what you can see is that the traditional learning algorithms, so those are those algorithms that were under the machine learning grouping, but not under the deep learning grouping, they tend to plateau with more data. So you give them more data, and then the performance doesn't necessarily increase any further. Whereas what's interesting about deep learning is that people have found that the more data you give deep learning algorithm, the better and better the performance tends to get. So people have tried with 300 odd million images in a Flickr data set, and they found the algorithm tended to become better over the course of the training. And so that's what's exciting about deep learning, is that it tends to improve with the more data you give it. And so over the last few years, there's just been this big kind of confluence, or this bringing together of having enough data to train these algorithms. And by data, I mean images of, say, cars or coffee cups or people. And you have enough data because people are taking photos of lots of things and uploading them to Facebook and tagging their friends. And that's been combined with these new algorithms. And then that has all been enabled to run in real time and to be trained to these really complex, large algorithms because of the advancements in computing power as well. And so that's why everyone's really excited about deep learning, because all these three things have come together to make it actually function properly. The idea of deep learning, I guess, has been around of using artificial neural networks has been around for some time, but it's that everything coming together now that has really allowed it to function. And so I thought I'd go into what computers really see and try and give context around the difficulty of weed detection, because while there is this hype, it is a difficult thing to confidently detect weeds. And so while humans have the capability of seeing an image, here we have an image of ginger, and we can identify perhaps different leaves or the background soil over here. But what computers really get is just a grid of numbers. And for a standard red, green, and blue digital camera, the computer gets 3 different color channels. So these numbers will be replicated for every pixel 3 times. So you have your red channel, your blue channel, the green channel, and the blue channel. And those intensities are typically between 0 and 255. So a really bright white area of the image is probably going to have really a high reflectance in all of those channels because it's white. A darker area might have lower reflectance or lower intensities. And what we can then determine is calculate things like edges or different types of features that are in each of those images. So I zoom on a part here, you can see that the darker area has lower numbers and the brighter area has high numbers, because that represents that intensity of the light that's being intercepted by the camera sensor. And of course, green areas would obviously have more intensities and higher intensities in the green, and then blue and red. If you have blue and red areas, they would have higher intensities in those different channels. And this is what a computer sees. It's not intuitive, and that's why you have to give it lots of data so it can start to identify patterns in these numbers. Because at the end of the day, an image is just a set of numbers for a computer. And the way we teach that computer for the machine learning process is with this image annotation process, which is hopefully what you guys might be able to do with some of your information and images later. And as you can see, this is tedious. It's you have to go through every weed, every image that you collect, and identify which part you want to detect. So in this image, we're trying to detect or annotate ryegrass growing points, because grasses are quite difficult to annotate given how spindly and small those leaves are. But here we're just trying to pick out all those different ryegrass plants in a sparse wheat crop. So this is me doing the annotation a couple of years ago for a data set that is actually now available on Weed AI if you want to download it. But that's the reality we're facing. So that brings us to the barriers to development of this technology. There's all the hype, there's all the promise, but what's actually holding it back? Why can't we have it tomorrow, for example? And the reason why is that you need large quantities of very high quality images. So I mentioned they trained that algorithm on a 300 million image Flickr data set, right? And that's where they continue to see algorithm improvements. And imagine trying to get that for agriculture. It's difficult. So all the images need to be high quality and annotated. So you have to actually have the weeds identified in the image so that the computer can start to identify where those weeds are and look for those patterns. It needs to cover all the weeds. So if you wanted to go out into a paddock and identify every single weed that might pop up in your paddock over that time, then you need to have a data set that covers all the possibilities, right? And the environment and the crop conditions also change, soil color. So this is from Texas A&M University's farm. And as you can see, the soil is very red. But if you go up to near Geraldton, Nickers, then you can have white sands or even yellow sands up near Una, or over in Narrabri, it's very dark, rich sort of black soils. So covering all those different soil colors and types is obviously another challenge too. And then the growth stages of the crop, growth stages of the weed, and of course the seasonality of agriculture. So we only have 1 year or one chance every year to really pick out those each of those different growth stages of the weeds at each of the crops. And things like growth stages, well, are these two weeds really the same? Of course, that's palm amaranth and cotton, but this is a very young weed and this is a large weed. But if you taught a computer to look for these plants here, it doesn't seem logical that it would be able to detect these weeds here, considering how different they look. So how do we try and incorporate that variability in agriculture into our data sets? And so, well, that's where Weed AI comes in, because it means that people can collect photos of images or weeds in certain scenarios that they want or have available to them, and then can upload it for everyone else to use to make their data sets more diverse too. And so it really takes that development of these algorithms and the data sets to the community. So it's a really community-driven thing. So as I go into Weed AI, I'd just like to say that this is a project that was developed or funded by the Grains Research and Development Corporation in Australia, GRDC. And it's very much like a big team effort from a range of different people. So the University of Sydney led the project. Dr. Michael Walsh, who's on this call, he led the project, and we had collaborators from the Sydney Informatics Hub and also the Department of Computer Science. So they've done a lot of development behind it, and it's been in consultation, I guess, with us as weeds people to make sure it fits the right specifications. And of course, we had a big pharma and also different researcher collaboration as well. But I'd like to thank those people too that really put the effort into developing this. I will just leave it a time if anyone has questions about that background first, though, before I start on to the live demo. Is everyone pretty comfortable with that green on green backgrounds?
Yeah, guys, there been any support from the commercial players? Like obviously Bilberry would have a massive database of images, and I don't imagine they're going to go and upload them onto the Weed AI website, because they'd be giving all their competition a big leg up. But has there been any sort of support or any interest from any of the commercial players that they might get on board or contribute something at all?
So Bilberry, AutoWeed, and a group in Melbourne called Altera. Liam Burns is on Twitter. He often talks about them, but they contributed, I guess, in that discussion points during the development, I guess, of Weed AI. They gave us some feedback. They weren't actually on the project, but they just gave a presentation, I guess, on how it might benefit them and how they're using, I guess, green technology. The difficulty is in, as you mentioned, getting people to upload or commercial groups to upload images, given that there is value in having large annotated data sets, as you'd expect. But like, it's something we're keen on having commercial people contribute to, even in small amounts. So whether or not that's just say 100 or 200 images just to help the community out, it's certainly a valuable thing to the community, but might not necessarily have that same commercial value to the company. So something we're very interested in having those groups contribute, but of course there are very understandable reasons why they may not.
Yeah, no worries.
Awesome. All right, so feel free to jump with any questions as well over the chat as well, or as they come up as well, and tell me to stop or slow down if that helps too. But at this point, it's probably a good idea to open up a browser if you're on a computer, or if not, just listen in and follow along on my screen. But we're going to head over to the Weed AI website now and just take you through some of those the components of that, and walk you through just what you can do and how you can play around with it. But if you want to head over to it, it's weed-ai.sydney.edu.au. So I'll just flip across to that. So hopefully everyone should be able to see my browser now. And when you open up Weed AI, so you go to Weed AI, you'll probably end up on the About page. So this is the home page for the platform. And that just tells you about the platform itself. So why do we have Weed AI? I've explained some of it in the presentation already. How is it different to what's there already? Because there are some publicly available data sets. Weed AI is all about being able to contribute as well as download too. And then a bit of background about navigating it, some of the different data formats, which I'll get to, and of course, acknowledgements, which I've talked about too. You can also contribute on GitHub. So for those of you with some software or programming backgrounds, the entire repository is also available on GitHub. So we click over here, you can find everything you need, where all the code that actually supports the website that was being developed by Joel, Henry, Jichao, and 11 that actually makes this website run. And there's a huge amount of code that goes into making these things look effective. So yeah, massive thanks to them who are working on that. But we'll go back to the Weed AI and head over to the Explore tab. So if you go to the top left-hand corner, and you should now come across a series of images that have been presented to you. So this is a random selection from the data sets that have already been uploaded. And as you can see down the left here, we have 4 different crop types at the moment. So we've got chickpeas. These were collected up in Narrabri. Lupins, these were collected in Western Australia, in Geraldton. We have some pasture images. These are from rangeland, I guess, pastures, collected by Alex Olson for his PhD. And that's called the Deep Weeds data set. And then also some wheat images as well. So say we want to filter by, let's go with wheat. So we click on the wheat category, and that will bring up all the images that were collected in wheat crops. So we have some with Lowland richardom, which is annual ryegrass in Australia. Over in the US, it's known as rigid ryegrass. And these, if you hover over that red box, you should be able to then see the entire taxonomy for rigid ryegrass. So you can see it's Lolium rigidum down the bottom here. And this is all developed through the GBIF database. And what it does is looks at the Weed Science Society of America composite list of weeds, and it pulls the common name from that. So that's why common names are often really tricky to go by, because they change depending on where you are in the world. So that's why we always present the scientific name first. You can also see that it's a bounding box. So you can see the red box here. And then it's in the wheat crop at a seedling stage. You can see the next image across is these might be in different orders depending on if you're in a different site or different randomized pattern. But you might go the next image across, and you can see it's a wild radish. So turnip weed, Christian ricosum. There's a wild radish further down. But turnip weed is a problem in the northern New South Wales or northern growing region of Australia. As you can see, that taxonomy again. Say if you like this data set, so you want to download the turnip weed, then you can click on See Data Set just below. This is booting to your emergent stage. It's slightly older than these images here. Click See Data Set. That will take you to this data set here. So this is what I called it. It's taken in 2020 in October, 14th of October, and it's at Cobbitty, which is a town just south of Sydney. And here you can see a description of the data. You can look at all the different images or just a selection of them, 101 that are available. And you can also see a statistic overview of what's available too. So we also have Lolium rigidum. So we have annual ryegrass in these images, as well as turnip weed. We see there are 55 images with Lolium and 71 with turnip weed. And all overall, we have 94 and 174 or 168 or 268 bounding boxes or instances of each of those weeds together. We also have information on the crop type. So wheat, we've got the BBCH growth stage, soil color, surface cover, percent surface cover, and also the location. And then also information on photography. So this was taken with a FLIR, black fly. This is the model of the camera with this lens, focal length at this height and this angle with a field of view. There's variable in this scenario, but it could have been fixed depending on how it's collected. And also this is collected at night with artificial lighting, as you might have been able to tell. And finally, this is a description of the photography and just general other details as well. But that's the dataset page for every one of these datasets has been uploaded. And if you want to download these datasets, all you need to do is click on top right here. It says download in WeedCoco format. And that will give you a nice download zip folder of everything you need, everything you want. If we click on datasets again, so this will take us to the datasets overview page. And this is where we have every single dataset that's been uploaded that's available to view on that explore page. So as you can see, this says the upload date, dataset title, and also to explore the images. This will take you to that main page, but it's got that filter on for the dataset you want. So this is a northern WA wheatbelt blue lupin dataset. So every blue lupin leaf has been annotated. And as you can see again, Lupinus cossentinii, you can see the taxonomy here too. Lorenzo just asked if you can add other crops, and you certainly can. So what I can do is I'll just go to the upload page. So if you scroll down to the bottom of the upload page, and where you specify all of this, I'll get to it a little bit later as well and describe in more detail, but it's called the ag context. And this is something we've developed to try and represent all that variability that I was talking about before in agriculture. And so on the ag context, this is the metadata you collect when you're collecting imagery, so you can then share those images with other people, and they understand the agricultural setting, like the environmental conditions, all those things that I've been through already, and they can understand which ones are available or which ones aren't. But as I mentioned, these are different crops you can add just with a drop down. So these are all the GRDC crops. So in Australia, we have very specific crop groupings for each of the funding bodies. These are the ones here, but you can also add other crops too. So if you type in a scientific name, they say for caret. I think it's, someone correct me if I'm wrong, it's August rota, August potentially. And then that will look into that GBIF database and then find the caret or whichever name for that crop is common. So you can also add different crops to it. You also add nodding crop as well. So if you're in pasture or fallow situations, you can do this as well, because that's quite relevant if you're using collecting images over fallow fields. But this is the ag context information that you can include when you collect data. And these are from soil colors, surface cover percentage type. We have surface coverage percentage, location, and also your growth stage here. So it's in the BBCH range, which is a more widely recognized growth stage range, as opposed to Zadox, I think, if you're familiar with that in Australia or potentially elsewhere. Or if you don't have a growth stage for the crop, so if this is not collected in the crop, which say in the case of the fallow scenario it wouldn't be, then you can just click not applicable. And then you also have a spot to add all your camera make, lens, focal length, height, angle, field of view, the ground speed at which is collected, lighting conditions, and description of the photography too. Finally, we have some information back on Weed AI now. So heading over to that Weed Cocoa tab. And this is the annotation format that we've developed that is meant to incorporate the COCO, so the MS COCO or Common Objects in Context format, and as well as that agricultural contextual information. I'll just detail that a bit further later. But this tab provides more information about that too. Next week, so in 1 week's time, I'll go through the process of uploading. It's a bit too much detail, I guess, to include in the 40 to 40 minutes to 1 hour I've got today, but I'll go through this in more detail next week. But if you just want to play around with this, feel free. You can search by any of those terms I've highlighted previously. So you can search by species, you can search by family, so you can find all your Asteraceae species. You can search by growth stages, locations, so you could search up Narrabri or Sydney to find the ones we've uploaded, bounding box or classification type tasks, and then of course the artificial and natural lighting as well. That's Weed AI. So anyone have any questions or want me to go over anything with that again? Okay, so I'll continue on, but feel free to. Rem's just added regarding the weed common name. So that's taken from the Weed Science Society of America. That's taken from that list. We try to avoid weed common names just because they do vary by country substantially. So scientific name is the best way to go. And yeah, the BBCH scale is what we've used for growth stages. But we use the GBIF database for all our naming and taxonomy. Apparently that's meant to be kept up to date as things change or develop. So that's what we use for taxonomy and hierarchy there. And yeah, EPO codes, I think, as well. I can get back to that if you want. I think it should also all be in apps. We highlight a. So if you click on the taxonomy, you can find where the GBIF species that it's come from, the kingdom, I guess it's come from. So Nick just asked about uploading straight from phone cameras, and because they need annotation first, you will need to upload it to your computer and then annotate it on your computer and then upload from your computer. But probably theoretically, if you had all the everything on your phone, you could do it from your phone. Although it is, I can't say I'd want to go through that process, so I wouldn't recommend. I think it does have to be done from the computer itself. So Mike's just asked about uploading images and having latitude and longitude embedded into those images if it will be captured. So this is something we did discuss, and we went down the path of removing all EXIF data, so all that metadata that's attached to images during the upload process, just to make sure people didn't accidentally give away locations. And we also went and made it the location information on a data set basis. So rather than a per image basis, having the whole data set located to a region is probably an easier way potentially of making sure that those images are in one defined area. Someone has raised that before, and it's something that would speed up the process of uploading, I guess, if you're doing it from mobile phone photos. But what we'll also come across as well is, say that the cameras that we use don't have any embedded GPS data on them. So we have to add it separately anyway. So I guess that's been our experience, is by adding just a single value for the whole data set that's collected in one field as opposed to individual images. So there's probably pros and cons of both at this stage, at this level of development. It's one of those features that could be added in the future if there was that continued development and funding. But at this stage, we've just rested with that single longitude and latitude per data set. But it's a very good point. Something that you can see the pros and cons of both. So it's something we would consider. But thanks for those questions. Feel free to, I hope I've answered those. Feel free to jump in if you have any more or want any specifics. I'll just continue with a little bit more information on Weed Cocoa, and that might help answer a few questions too you might have. So Weed Cocoa is the standardized format we've developed to try and capture that agricultural context information with this annotation format. So when we try and train an image and develop these data sets, we have to have a consistent format with which we give that training information, those bounding boxes that you can see, to the computer and to the algorithm itself. And so we've chosen common like Cocoa format. So there are a range of different formats you can choose, but Cocoa is probably the most flexible. It also only has a single file per data set as opposed to multiple files. And there are many existing tools for converting it between formats. So say if you have a YOLO model, which is a you only look once model developed in the 2016, I think, 17, the version 1 anyway. They use a different format to Cocoa, but there are some converters available. But it's widely used and developed by Microsoft, so that helps. When you combine it with the ag context information and also metadata surrounding the authors, who's collected it, the name of the data set, the licensing information, that's when you end up with the Weed Cocoa. So that enables us to then search it like we've already done, and then it's called indexing. So we can search through that data and then filter out what we need or what we don't need. And to develop that yourself, if you just head along to, you can go to the upload page, and you can click on construct and save an ag context, or construct and save metadata. And so these are the two forms you need to complete. And you can add those files at the time of upload. But those are the links to them as well. So the data standards we developed are something that helps the people report everything consistently. So this is the ag context editor that we've seen, and it helps us evaluate model performance if we have, say, data gaps or weaknesses in our models. So we can then filter by, say, red soil if the algorithm doesn't perform on red soil very well, or say it doesn't perform well in a grass crop, or sorry, early growth stage wheat crop. You might be able to then filter for later growth stages or early growth stages to try and work out and download data that can fit that model. It also obviously gives us the searchability, so indexing, and helps with model generalizability. So it's how an algorithm or a model can work in different environments if it's trained on one set. But by having everyone able to upload data and also tag it with these contextual information, then we can start to help and understand and improve that generalizability to different environments. And also interoperability. So say I develop a data set, Nick develops one in Australia, Lorenzo does one in Chile. If we all report the same information in the same way, then all these data sets can work together for these different models. So we can collect them separately, report the same things in the same way, and then they can work together. And finally, for benchmarking too. So if we want to develop new algorithms that can be tested on the same playing field, we need to have them in the same conditions, or at least understand which conditions are relevant and available to us. These are the different standards that we report. So we've been through a little bit of these already. So we've got the crop metadata, environmental metadata about the location and weather, the camera metadata, so things about the make, lens, focal length, height, angle, field of view, and photography description, and then also just broad image collection metadata too. So it's been cropped to a plant in the case of, say, images that have been collected in a glass house, or things that have been cropped to that individual plant from a larger image. You can tick that box too. When you combine all this information with the annotations in that whatever format you have, or COCO if you've annotated with that, along with that data set info on who's authored it, then we end up with WeCOCO. And so that's what WeCOCO is all about. So it brings together these standards, the standard annotation format, data set information all together into one file. So that's on the WeCOCO front. Any specific questions on that you'd like me to recover or go over? So Brad just asked about focal length being extracted from phone images. Again, that's metadata that, like Mike mentions, that you could potentially extract. I know some phone cameras, like my Samsung, reports the focal length and the lens model. So that could, again, be extracted if we kept EXIF data. But for privacy reasons, we defaulted initially to removing all EXIF data from images. But say if you are developing, if you go out with your phone tomorrow or during the week and you do collect image data, if you right-click on those images, select properties, and then scroll down through those image properties, you can find the focal length of that lens that's being used. But I guess to answer your question, Brad, no, focal length won't be extracted in this method, but it's something that could be developed. So if there are people interested in contributing to the website, because it is open source, these sorts of tools could be added, I imagine. If someone knew how to do it, probably fairly straightforward. So to collect images is the next step in the process. So we've been through the data formats, we've been through with AI and also why we need it. But how do we actually do this? So this is the practical side of things. And this is what would be great if you have the chance over the next week or so to collect a few images and test it out for yourself. But the first and most important thing with image collection is really having a goal in mind. So what are you doing this for? Why are you spending your time doing this? And it might be for weed identification, for spot spraying, or from a different platform. You might need an angle. You might want to have straight down, as in these photos here. And you also want to understand which weeds you're trying to target to. Are you trying to get them in fallow? Do you want to get them in chickpea crop, as you can see here? Or is it better to have them in wheat crops or whatever you have available to you? It could even be your own grass or your lawn at home, if that's the closest thing. And the next part of this step is have consistent parameters. So as with the ag context, you need to be able to report the camera type you use. So you don't want to try and combine two cameras into one data set. You need to have the height consistent. So here we have a backpack system we've developed at the University of Sydney. So that keeps our camera height relatively consistent, and width and angle is consistent as well. Or you could equally go out, as is quite easy to do, and just hold your phone at a certain height. So for me, if I hold my phone about straight out from my shoulders, it's about 1.5 meters. And if you go out during the next week, you can do the same. Then it might also be a set height. But as long as you keep that consistent and that angle consistent, that's what it's all about. And then also the lighting too. So to report or collect a data set during a specific time of day, sun conditions or cloud conditions, that's also important. So that when people search through the data set, they get consistent information returned to them. So that's when you're out in the field, or if you can remember, record that metadata with the ag context editor or write it down on a bit of paper and then build that file later. The Add Context Editor does work on your phone, and you can actually save that file to your phone as well. I'm not sure how that works on iPhones, but I know with Samsung you can access those files, and with Android. And finally, make sure you save and obviously back up those images too. But that's probably the majority of information you need for image collection. So as I mentioned, over the next week, see if you can collect, say, 10 or 20 images of a weed in a certain scenario. Some of you might have access to crops. Some of you might just have weeds in your lawn or your maybe the local park. Have that consistent environment, have a specific weed in mind that is a problem, and record that metadata, annotate them, as I'll explain shortly, and also then we can start uploading to Weed AI. And next week is when I'll take you through that process in detail. So image annotation is the next part of this process. You've collected the data, and then now you need to actually develop that data set, and that can then be fed into a machine learning algorithm, and then that machine learning algorithm can try and recognize the patterns that are present in each of these annotations that you can see here. So this is where we generate that ground truth data that we can test against to see if it's actually learning and doing its thing properly. So the important thing to note is that an algorithm is only as good as the data you give it. If you give it bad data, if you label the wrong thing, or if you mix up, in the worst case, I guess, is mixing up, say, the crop with the weed, then the confidence in detection might drop off dramatically. So it might start detecting the crop as a weed, or it might not train very well because that diversity is quite large, and so it can't try and fit an algorithm to match that diversity. So, for example, here we have a sow thistle, and potentially I think we had turnip weed in here as well. And if we label the sow thistle turnip weed, then the algorithm starts to get confused between the two, and either it doesn't train at all, it won't finish its training, or it just performs poorly and might detect the sow thistle as turnip weed, and vice versa. Just make sure you get that data as high quality as possible. More data is always better. So more images, the more instances of weeds. So I guess there are two factors here. It's the number of images and also the number of weeds in each of those images. And as you've probably seen, this process is quite labor intensive. There are a whole range of different annotation tools available to us. This is a list of most of them that I could find. I think there are a few more that I've missed out, like RoboFlow. But the ones that I've used that are probably the best are the computer vision annotation tool. This is developed by Intel, and that gives the right output format that we can use for the Weed AI website. So I'll send around an email after this, a link to all the relevant tools you might want to use, and also some walkthroughs of each of those annotation tools and how you can use them. But there are a lot available. I tend to use Label Image, but that does require some command line and terminal and a bit of coding experience, I guess. Whereas Computer Vision Annotation Tool is an online website. You can just upload your tools, your images to, and use the tool as you need. And there are a few tips, I guess, you can follow that help the process and, I guess, improve your output. This is taken from the RoboFlow blog. So RoboFlow is a website that tries to talk you through the deep learning and detection process in a whole range of different data sets, so not just images, but sound and video too. But the most critical point is to label every object in each image. So if you're looking for all the wild radish, make sure you've labeled every single wild radish there. If you forget some, then it starts to think that that's background and can train against detecting it. So it's really important that you label and you get every single one. Have specific labels. So for this, we want species-wise. So you need to have, So for this one, you need to have the species level annotation. What we're also exploring is if growth stage annotation is necessary to improve it. But at this stage, we've just found that species are probably the good starting point. Make sure the bounding boxes, if you're labeling with object detection, which I'd probably recommend you do for this data set, make sure you label those or have those bounding boxes quite close or as close as possible to the edge of the weed, as you can see in some of these images here. So it's right up against the edge of the weed, but make sure you don't chop any of the weed off. So leave a few pixels gap. Annotate any occlusions. So if there's a weed overlapping another weed, make sure you annotate the entire of that overlapped weed and do both of them if they're both of the same species. And be consistent with the way you go over an image. So if you start in the top left and then go down one corner, move across some percentage, then go up, and then go over that image in a serpentine fashion. And that means you won't miss any parts of that too. So that's the annotation process. Anyone have any questions about that, the image collection or annotation? As I mentioned, I will send through these links for you after this in an email. So feel free to go along. They have some great videos and some tips, I guess, on how to annotate too. So I'd just like to finish up here on saying that green on green spot spraying, while it seems like the obvious first step, it's really just the beginning of this detection technology, because we can start to use it for laser weeding or targeted tillage or a whole range of alternative methods that really aren't practical in conservation cropping systems. If you end up plowing the whole field, in the case of targeted tillage, or lasering the whole field just isn't really relevant. If we can deploy some of these really powerful detection algorithms, we can start to use some more alternative techniques that can either complement or replace herbicides depending on the way you're controlling your weeds. And also, I'd just like to mention, while we've been developing Weed AI as an open source data source, the Open Weed Locator we just recently launched this week. And this is a open source software and hardware green on brown weed detector. So it's image based, and what it does is just pick out that the green color of weeds in fallow situations. This isn't no machine learning involved. It's just, I guess, artificial intelligence or whatever you'd like to call it. But people can go check out the full code. They can build one themselves. And that link is down here. Just if you search up Open Weed Locator, or you can add to this GitHub link. Feel free to check that out. And then it comes down to, well, why do we do all this? Why do we make the effort? What's the point? Well, it's all about these opportunities we get for reduced weed control costs, inputs, the ability to control weeds in new ways, and what that really opens up for us in terms of agronomy and managing our crops in different ways. So I guess that's what weed control is all about, is being more effective in the way we manage competition with crops. And if weed detection enables us to do that more efficiently and opens up new opportunities, then it makes sense to invest in our time and effort and energy in trying to improve that for a whole range of people to use as well. So I'd just like to say that, I think this is the wrong slide, but anyway, next week, 2nd September, 10 a.m. in Australia, if you're in the East, or 7 p.m. on the Wednesday night, if you're in the US, in Texas. But thanks very much for joining in. Just like to thank everyone involved in this project. I guess I'm just a mouthpiece at the moment, but there's a massive team behind it all that certainly makes this all happen. So thanks to GRDC for funding it, and all the people involved in this project as well. So appreciate everyone being involved and coming along this evening or this morning, depending on where you are. And feel free to ask any questions if you have any at the moment. Also feel free to email me or contact me on whatever platform you have me on. But are there any questions or burning questions that people have at the moment? Brad's just asked about how many images or weeds to get something to work. So it's a very good question, and it's a hard one to answer specifically, just because it all depends on, as I said, both things, on the variability of that data set or the weeds you're trying to get detect. So if you're trying to detect in very consistent conditions, environments, you have controlled setting, then you will need less than, say, if you're trying to detect over 1,000 hectares of a wheat field across all the seasons and all the different types of lighting conditions, day and night. What I often work on is just a rough guide I try and do to myself, is using about 1,000 instances of each weed or 1,000 images roughly to try and get some form of detection that can work somewhat in a field. So I work around 1,000 figure, but that's by no means enough that would be useful to deploy something commercially or any of those things. It's just a figure I like to use myself.
Hey, Guy, I'm not sure, hopefully you can hear me, but Alistair Clark here from CSU back in Australia.
Yep.
Yeah, thanks for the presentation and yeah, I'm really interested in I think in the part around data standards and making sure there's that interoperability. And yeah, obviously that's been well thought into with your research, which is great. And I hope at Sydney Uni, they're going to get some of the undergrads to do some image annotation for you guys. Be a good one for them. But I had a question around like whether this can be applied, thinking of it the other way around, where you're actually annotating the crop that's meant to be there. And if you have the crop being annotated, and I guess knowing what the soil is, or maybe having to annotate the soil if there's an option to then spray or laser or precision till what's not meant to be there.
Yeah,
if that makes sense.
Yeah, for sure. I think it's a really good point. So one thing, to me, that's a lot like fraud detection or spam detection in emails. So it's like it's a big field of study where you have, say, 1 million emails are right, are the right ones. You want to keep them, and then 1 or 2 emails are spam, or potentially that's more relevant to fraud cases. Because I think there's so much spam these days. But it's where it's more often that you detect the outlier, you detect the anomaly, which is anomaly detection is what they use. And in this case, it's probably what crop, if you look for the crop, you look for things that are different, that it could potentially work. What deep learning has trouble with, I guess, is if you don't provide it clear boundaries on what to detect and what to avoid, at least for image-based things that I've worked with, then if there are no clear boundaries on what to detect and what to avoid, it can sort of lose performance on identifying those outliers. It's something I'd really like to look into, I think, is that outlier detection in a weed and crop scenario, because it's a question we often get, is about looking at the crop and then looking for things that are different to the crop. And I don't think there's probably no really good answer about that. Where it might be relevant or more useful is where you have, say, a segmentation operation, and you can, or like a row detection. So for cotton, if they're really clear rows, you can identify the rows, cut the rows out of the frame, and then look for anything that's green in between the rows. And that's a good way potentially of finding weeds as well in that sense. But yeah, that's probably hopefully the best answer I can give, I guess. With lots of this stuff, it's fairly new, and there's no often clear answers at the moment.
Yeah, no worries, makes sense. Thanks, Scott.
Cheers. So there's a question from Lorenzo. So could you name 1 or 2 weeds, crop situations you need to work on soon? And perhaps we could advance in one of those and share on the platform. It's a really good point, is to focus efforts on particular species. And in Australia, there's a few that I'm working on, ryegrass in wheat. But I think what it would require potentially is having a list or a discussion, and telling people these are some that might be available in your area, or that people can contribute to. So it might require a little bit more thinking, but I think that's probably a good idea to have that direction, so that if people find the site, then they know what to start looking for. Do you have any, Lorenzo, in your experience, that you would like to see done? Like what are some challenging weeds that you have in Chile? Are you there, Lorenzo? Do you have any weeds you're thinking of, or?
Can you hear me?
Yep, hear you now.
Yeah. Here. Hello to everyone. Well, here in Chile, we have a lot of crops and weed situations. We have a variety of, for instance, we have with the same situation that you are dealing there in Australia. We have here lolium in wheat. We have you don't have this perhaps in rice. We are working a lot with echinocloas in rice. This is one situation that you mentioned in your exposition about one grass over other grass. That is very noisy here. and the others are broad lists on other broad lists, for instance, in pulses, in lentils, in industrial crops like sugar beet, etc. We have a lot of situations. For this, I am telling you that perhaps if you need more one particular situation, we could put images on those in your platform. We are working currently in several crops and just trying to put those models in function in order to, for instance, with YOLO and etc. Well, but we can collaborate. This is I by the way, I congratulate you and your professors for this initiative. This is very nice initiative, and well, we are attentive to any requirement. If we can
right, I
collaborate.
appreciate it. Lorenzo, yeah, I think that the point of having targeted species is a good way to go about it. So then at least we know, or at least having that option there so people can contribute and develop a larger number of images more quickly for that one that we start to develop more relevant algorithms. Like probably if I'll send you an email potentially afterwards if you'd like to, if you have some images in mind or scenarios in mind. I know personally for me it's ryegrass and wheat, but
I have some data sets in the my workflow in the last 6 months or so. We have some few data sets in we are putting the bounding boxes in the supervisory, for instance. And after we are working in RoboFlow, for instance, and well, but in the future, trying to deal with the requirement for the collaboration, we can make the tagging in the CVATs or any other that you need. Is possible.
I think RoboFlow or supervisory will output, as long as it's that COCO or Pascal VOC format, then they'll be that'd fit with AI. Do you know how many images or the data sets you have available currently? Like, are there some you'd like to just upload?
Yes, I have. I can send you afterwards if you want. I can share some folders by Google Drive, for instance, and because I was trying to upload this some hours ago, and there was some message is incompatibility between the scientific name and not the common name, the scientific name. There was some issue, but I was just approved. So I was trying to convert from supervisory and in RoboFlow to try to download in the COCO format, the COCO or in the VOC format. But we can work on that. For
sure. I'd be happy to talk you through the process. Yeah, for sure. Anytime. It's always good to get more people contributing. So that'd be fantastic. I'll put my email on the comments. That's my email there. So anyone see online can feel free to email if you have the questions. And likewise, Lorenzo, it'd be great to hear from you about you'll be going to.
Okay.
Yes,
Guy, just a quick question. I've been coming from a fairly low base, having never done any image annotation before. Like, obviously you'll send out those the relevant links to software packages. Just thinking, is there any that actually go through and I guess do a first run at putting bounding boxes on by color or something like that? Like, say in a bare fallow situation where the species that are there, you go through it, it does that, and then you go through and refine those bounding boxes. Just thinking about that annotation process, just wondering, does that even happen, or is just start from scratch every time as a completely manual process for putting the bounding boxes on?
Once you have a model trained, then you can start to do that correction process. So if you say, all right, apply all these bounding boxes to wild radish, and it detects them, and most of them are correct. There are 5% of them wrong. Then you can just go back and correct the 5%. That's a process that's used. There are none, and something that I know Daniel and I are working on is that color-based annotation initially. I don't know of any platforms to do that automatically for you currently, but it's reducing that annotation time is it's a massive driver at the moment for a range of different people. So I wonder if there will be a few tools come online in the near future that would help on that front. Because it takes a long time to annotate otherwise.
Yeah, I can imagine it does. And yeah, I mean, obviously I don't want to end up training it on the wrong things. Like, you still need to go through and make sure that it's right. That's why probably the manual process is probably the safest at this point.
Yeah, I think at this stage we're also doing, like, even with some of the automated tools are developing, you have that ground truth manual expert labeled data set just to double check. Otherwise you're not entirely sure what the ground truth is. So I'd recommend just this, as well as a good exercise, I guess, just to see what the process is like. Just have it, only do it on, say, 10 or 20 images. So hopefully that's probably 50 odd weeds or something. So it shouldn't take too long. And just have a go, probably do the manual way. There's CVAT, there are a few videos on how to use that. They'll walk you through that process on how to draw the box and how to annotate. And usually it does take a few tries to get it right, but yeah, so we certainly appreciate it if you could do a few.
Okay, we'll give it a go.
Awesome, thank you.
All right, thanks.
I think we're probably getting 15 minutes over time, but obviously you're interested, so if there's any more questions, happy to take them. Otherwise, I've put my email down there, so feel free to email me at any time, and I'll try and do the best to answer that. And for sure, feel free to send me find me on Twitter, or find WeedAI on Twitter as well, and get involved with that. But looking forward to chatting with you all in about 1 week's time, and seeing some of those data sets. And if you haven't had time to make one or annotate one, that's perfectly fine. I'll take you through the process anyway, and just show you how it all works. But thanks everyone for your involvement. Thanks for the feedback and the chats, and all the best with your work. Thank you.
Thanks, Guy.